So this is chapter 14 in the Raven textbook, DNA, the Genetic Material. And to start off, we're going to take a look at the history of DNA discovery, essentially. First, what is it that the genetic material, so in order for heredity to work, what does the actual physical, chemical structures have to be able to do? Okay, number one, they have to be able to carry information. So essentially, you have to be able to have the blueprint for an organism contained in this information. Number two, it has to be able to replicate. So it has to be able to copy itself, and it somehow has to be able to make new copies or somehow come together and make new organisms. Number three, transmission. So it has to be able to be moved from one organism to the next. In essence, if you're talking about sexual reproduction, it has to be able to come from the mother and the father. It has to be able to go from one organism to another. And number four, whatever the genetic material is, it has to allow for variation. The example would be no two human beings look alike or are exactly alike. What is it that makes individuals different? So variation has to be possible with the genetic material. Now, if you go back to the late 1800s, that's when they started to really come out with some of the biochemical laws of heredity. This is before the rediscovery of Mendel's work. And during that time, people that were researching this were convinced that chromosomes carried the genetic information, because just as we saw in lab, you can see chromosomes with a light microscope. Now, from the 1920s to the 1940s, due to different experiments that were being done, scientists thought that it would be protein that would carry the genetic information from one organism to the next. And this made sense because there is a wide variety of proteins out there. Think of all the enzymes, just the different kinds of proteins. It's a much more varied class of molecules than are, say, carbohydrates or lipids. So scientists were fairly sure that it would be the proteins that would carry genetic information. So let's look at Frederick Griffith in 1928. And he did an experiment using mice and using Streptococcus pneumoniae, which is essentially the bacteria that causes pneumonia. There are two strains. So when I demonstrate this, keep in mind there's two different strains of Streptococcus, or S. pneumoniae. There's S, which stands for the smooth strain, which is virulent or disease-causing. So this is the one that can indeed cause disease in the mice and in people. There's also R, or the rough strain, that does not carry the disease. Okay, so they're non-disease causing. So what Griffith did is he infected mice, and he wanted to see what was the difference between these two strains. So if we take a look here, this is the live virulent strain of S. pneumoniae. So this is the S strain that is being injected into the mouse. And as we see, Time goes by. Mice die. Okay. Not good, right? Now notice here what makes the S strain is this right here. Think of this, and S stood for smooth. So it had a polysaccharide coat over the bacterium. Okay, now let's take a look at the R. Oops, sorry. To look at the R strain. And this is the rough strain. So in the rough strain, Griffith injects the mouse with these R strain bacteria. Mice are alive, right? So the mice live through this. Nothing happens to them at all. So essentially these are your controls, if you will. Okay, now we get a little deeper in the experiment. This is, again, the S strain. The S strain. And what Griffith did, is he heated these up to the point that they are, in fact, dead. So these are the virulent strain. They cause disease in the other mice that he injected them into. So surprisingly, or non-surprisingly, actually, with this one, he injects the mouse. The mice live. Okay, so he's demonstrated now that one strain causes disease, one strain does not cause disease, and the strain that causes disease, when it is killed, does not cause disease. Interesting. But now comes the really interesting one. He takes a mix of the heat-killed virulent strain. So this is the S. And he takes live, non-disease-causing, and mixes them together. So now think of this. Both of these together, or I'm sorry, both of these separately on their own did not kill the mice, right? If we go back and we look, the smooth strain that's been killed by heat and the rough strain, both by themselves, did not cause disease. But lo and behold, Griffith injects the mouse with a mixture of these two, and the mouse dies, okay? And he would autopsy these mice after they die and find out whether or not there were any bacteria in the lungs, because if they, obviously the mice die from pneumonia. So if they die from pneumonia, there's going to be pneumonia in their lungs. So any of the mice that have died in this have pneumonia in their lungs. Well, lo and behold, from this mixture, the mice die, and Griffith finds lung tissue that have the bacteria. So what does that mean? He called this, he didn't know exactly, but he called it transformation. So somehow the ability to cause disease was being transferred from the dead S strain cells into the live R strain cells. So the interpretation, if you will, was that somehow the genetic material or ability to cause disease in this case, was somehow transferred between the cells. And Griffith had no idea exactly what it was that was doing it. You know, you're going back here, this is 1928, so no idea. Okay. Now fast forward 16 years or so, and we get the experiments of Avery, MacLeod, and McCarty, okay, a team of scientists. And one of the underlying themes of almost anything that you look at through the 20th century especially is an increase in technology. In advancements in technology were rapid in the 20th century. So 1944, we're looking at near the end of World War II, and basically Griffith's experiment was done again. Okay, so it was repeated again with a little bit of a difference. Okay, what Avery, MacLeod, and McCarty did was they actually took and they were able to remove all the protein from the bacteria. Okay, they could remove all protein components, and it did not destroy the ability to transform those rough, non-virulent cells. But taking and removing the nucleic acids did destroy the ability of the bacteria to cause disease or to transform. So now this, mind you, 16 years later, was some of the first support for DNA as being the genetic material, or rather nucleic acids as being the genetic material. And I'll show you a little mock-up of this. So here we go, and again you're using mice to check this out. So here are these heat-killed S cells, right? So these are disease causing. And through technology, essentially, through better lab equipment, better technology, really, they were able to remove both lipids and carbohydrates from the solution of heat-killed S cells. Okay, so what remained in the solution was protein, RNA, and DNA. Okay, so there's protein, RNA, and DNA. All fats, all lipids, all carbohydrates taken out. Now, case one. Oops. Sorry. So, all right, here we go. So, number one, they add proteinases. So proteinases are enzymes that digest proteins. Thus, in this sample, there would be no protein. So what remains should be RNA and DNA, okay, but no protein. In number two, added ribonucleases. So these would digest RNA. So the sample here would contain no RNA. And number three are deoxyribonucleases. So these would digest or destroy DNA. So sample 3 contains no DNA. Now go back to number 1. Now we add the R cells. So remember, these R cells are no disease. No, they do not cause disease. and they add the R cells to this mixture, and what happens? S cells appear. So somehow they're able to transform in this mixture. Somehow the R cells pick up the ability to cause disease. So in this case, that is not because of protein, but it could be because of RNA or DNA. Okay, so let's go to sample 2. With no RNA, add the R cells in. And again, S cells appear. So they are transformed. They're transformed with no RNA in there. Okay, so it had to be either the protein or the DNA, except that this one had no protein. So it can't, and that leaves us with number 3. We add the R cells, and there are no S cells, or no transformation occurs. And of course, that is because there is no DNA. So if you read at the bottom, conclusion, transformation cannot occur unless DNA is present. Therefore, DNA must be the hereditary material. And this just a classic, incredibly great experiment. Okay, now fast forward another 8 years, and we're getting into the age of DNA. So at this point, researchers are pretty confident that it's DNA for the most part. There were still some out there that thought otherwise, but still trying to prove. Okay. Hershey and Chase, 1952. And they studied this virus that infects E. coli bacteria. And just FYI, a bacterial virus is called a phage, or a bacteriophage, just phage for short. Now, in a virus, they're outside here, as you see right here, the capsule and the whole body, if you will, of the virus. So I'll write here, virus. So the blue is all protein. And inside the head, or the capsid, is contained the DNA. It's a DNA virus. So again, this is a schematic drawing, kind of a science fiction-y, right, of a virus. But if you look at an electron micrograph of what these, so this is under an electron microscope, they actually kind of look like that. They sort of look like aliens. So it's called a T2 bacteriophage. So the genetic material is here, and to visualize this, by the way, the DNA is radioactively labeled. That's how you get this green color here. And in these, here you can see this DNA being injected right into the bacteria. So what were they doing? What were Hershey and Chase doing in their experiment? Okay, so follow along. In the first batch, the viruses, their phages, were grown with radioactive sulfur. Okay, radioactive sulfur. Oops, sorry. And sulfur is incorporated into proteins. So these viruses are bred, if you will, to have their proteins, or the coat, radioactively labeled with sulfur. All right. Then you take these radioactively labeled viruses and mix them with E. coli bacteria. What happens? Here we go. They inject the DNA into the bacteria. So now what they did after that, they allowed that to happen. Then they agitate the mixture. So they literally blend it, but not at such a speed that it would break up the bacteria, but at such a speed that the viruses pop off. So the viruses pop off, and they then centrifuge it, or they spin it. Now, when they spin it, you're going to get the solution with the viruses, because they're much, much smaller than the bacteria, separate from what's called the pellet here. And that's the bacteria and the contents of the bacteria. Okay? Now the question is, where do you find the radioactivity, right? Because remember, these were radioactively labeled with sulfur. So the radioactivity in this case is in the solution. So there is no radioactivity within the bacteria that were infected. Interesting. So now let's take batch 2. So these phages, viruses, were grown in radioactive phosphorus, and phosphorus is a component of DNA. So phosphorus labels DNA. Now what happens? Here we go. Put the viruses to work. They inject the DNA into the bacterium, right? Agitate the mixture. Viruses pop off, centrifuge it, and look and see what's in the pellet. Radioactive DNA. So what this showed was the radioactive DNA is injected. So the disease causing, because this is they're being infected. These bacteria are being infected by these viruses. So the infection causing, or the disease causing material is the DNA. Okay. Now we go forward a few more years in the 1950s, and they are hard at work in many laboratories around the world to try to figure out what is DNA. So working in a laboratory that was very close to where Watson and Crick were working is Rosalind Franklin. And she didn't receive the credit she should have because she was a woman in the 1950s. She also died in 1958. But she was performing X-ray studies of nucleic acids, in addition to other things. But she discovered, and she's now generally given credit for being the first one, or one of the first ones, to suggest that DNA was a helix. Okay, that it had a helical structure, or the twisted structure. In fact, she might have been not 100% sure to say that it was a double helix. And we can see her now famous, I believe it's called Photo 51, right here. So this here. Photo 51. So these are X-rays, and you're essentially looking down the double helix, and suggested that it had a diameter of only 2 nanometers, and that the helix makes a turn every 3.4 nanometers. Now this is extremely small, right? And no way you can even see this with an electron microscope. So we have no way of actually visualizing this. It's so thin, okay? Well, she is the first one to suggest that. Subsequently, Rosalind Franklin died of ovarian cancer in 1958. Might have been caused or aided because of her work with X-rays. So exposure to X-rays, nobody will really know. But she was not able to share in the Nobel Prize with Watson and Crick, and Maurice Wilkins was the third to share in that Nobel Prize, because the Nobel Prize is not given after death. You have to be alive to win the Nobel Prize. She died in 1958, and I believe they won the Nobel Prize in 1963 for the structure of DNA. And that's the next one. James Watson and Francis Crick. So using this evidence from Franklin, from Maurice Wilkins, from Chargaff, we'll get to him in a moment. They're the ones that came up with and built the structure of DNA. And they proposed the double helix structure, or confirmed the double helix structure. Important to know, they didn't actually perform a single experiment. They were using the work of others. I don't want to say, what they did was great, but they were not experimenters. They were theoreticians. So they were just working out what the structure was. They were on a mission to explain the structure, if you will. So they proposed the double helix, and from there, the rest is history. So now we look at the structure of DNA itself. And when we look at the overall structure of DNA, we have to go back and look at the structure of a nucleotide. If you recall, nucleotides are the building blocks of DNA, just as amino acids are the building blocks of proteins, and so forth. Okay. All right. So let's take a look here. Okay. So a nucleotide, the first thing that we have is our 5-carbon sugar, or deoxyribose. That's right here. Okay. So deoxyribose is the sugar that is in DNA. The next part is the phosphate, which is, if you recall, chemical formula PO4. And there it is, PO4. We also have attached a nitrogenous base, or just meaning containing nitrogen. So the bases all contain nitrogen. There's actually, here's a nitrogen, here's a nitrogen. And this is called a base. Now this is attached freely, okay, to the deoxyribose sugar. And there are 4 in DNA. The first 2 are called purines, which are adenine A or guanine G. Or they could be under the class of pyrimidines. Pyrimidines are cytosine and thymine, C and T. And we will see this is a pyrimidine right here. But we'll see on the next slide the different ones. Don't need to memorize the actual chemical structures. And the other part, if you will, is actually a piece. And that is the OH, or hydroxyl group, that is attached to specifically this 3' carbon of the sugar, of the deoxyribose. So now when you see this is a 5-carbon sugar, right? So you have oxygen up here. So this is a carbon, 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 and carbon right here. Okay, so that carbon is always going to be the one that attaches to a phosphate. And also we're going to see how the hydroxyl group right here is going to attach to the next phosphate. So when we take a look, again, we have our phosphate, our deoxyribose sugar, and then here are the 4 possibilities that we have for DNA. Here are the 4 possibilities, right? Adenine and guanine, which are purines. Now, it's important to remember that they are purines, not important to remember the structure. But it might help by knowing that you have a double ring with the purines. So you notice here there is a 6-member ring connected to a 5-member ring. Okay, so a 6 and a 5. Same with guanine, a 6 and a 5. You'll notice that the only difference between these two right here is the double bonded oxygen and the position of the amine group. There's an amine, an NH2 group that's on there. And the pyrimidines, or single ring, I always remember this, that they look like a pyramid. Yeah, this one looks like a pyramid, but it has something else attached to it. This one's just a nice pyramid. And the pyramids are T and C, thymine and cytosine. Good idea to just memorize those? Yes. Okay. Now, how are the bases, how are the nucleotides actually linked together? I shouldn't have said the bases, because the base here, right, the base, let me go ahead and turn the pen on. So remember, the base is A, T, C, or G. So in this case, by the way, this base, it looks like a pyrimidine, right? So because it only has one ring. So it must be, by the way, in this case, T or C. Okay, because those are the pyrimidines. Okay, so the bond, and what we're looking at is this section right here. That's called a phosphodiester bond. Okay, the phosphodiester bond. So you have the PO4, you have the phosphate group, right? And you see here's the 1, 2, 3, 4 oxygens. Now this is connected on this end to that hydroxyl group, right? Because look down here. Here's that free-hanging OH, okay? So you have that free-hanging OH here, which is now incorporated into the bond. And this oxygen at the bottom end is covalently bonded to carbon, okay? It's to the number 5 carbon. This is where we're going to get, or this is the reason for having this statement, which is going to become absolutely critical to know. The chain of nucleotides has a 5' to 3' orientation. That is referring to the 5' and the 3' carbons of deoxyribose. And you'll see this a little bit better, I think, on the next slide. because as we label these, 1, 2, 3, 4, 5. Here's the 5' end. So we have a phosphate. It's attached to the number 5 carbon of the deoxyribose. So it goes 5, 4, 3. This is the number 3 carbon of the deoxyribose, which leads into another phosphate, which creates this phosphodiester bond, which is then attached to the 5 carbon, then count down 4, 3, and the OH group is on here. And then that's where the next phosphate is going to come. So this is where the entire term 5' to 3' comes from. It all has to do with the carbons on deoxyribose. Remember, the name of DNA, or it stands for deoxyribonucleic acid. Okay. So next comes Chargaff's rules, which this preceded Watson and Crick in actually determining the structure of DNA. But Chargaff he experimented with nucleic acids, and he found out that in a species, in an organism, in a single organism, let's just leave it at that, the amount of adenine always equals the amount of thymine. Okay, so A equals T. And the amount of guanine equals the amount of cytosine. So G equals C. So there's always equal amounts of A and T and G and C. There's also, when you add them up, an equal proportion of purines and pyrimidines. So remember, your purines were A and G, and the pyrimidines T and C. So when you take G plus T, it equals C plus A. So let's think about that for a second here. Okay. The amount of adenine equals the amount of thymine. Guanine equals cytosine. But when you add G and T, okay, it always equals C and A. So it's kind of a cross. There's an equal proportion of purines and pyrimidines. So let's take a look at why this is. Okay, there's no need to memorize Chargaff's rules. So the DNA strands, in a strand of DNA, the nucleotides are covalently bonded, right? We saw that with the phosphodiester bonds, and there they are. Oops, sorry. So phosphodiester bond, right here. Notice, right? 5', 3', 5', 3', 5', 3'. And by the way, this leads to each successive nucleotide is offset a little bit from the one before it. So it creates like steps going down, and that is going to basically lead to the helical structure. So because it's stepping down, it's naturally going to want to twist, which is what it does. Notice that the bases here are not attached to anything. Bases not attached. Exclamation point, exclamation point, exclamation point. They're not attached to anything. Okay? So this leads to the backbone structure being phosphate sugar, phosphate sugar. So the backbone are repeating phosphates and sugars going down, with the bases sticking out. Okay? With the bases sticking out attached to the number 1 carbon on each sugar. Okay? Here's another view. So now we take it and we look at 2 helices. So now we take 2 and twist them together as the structure of DNA is. And we see 5' end, 3' end. Let's follow it, right? Whoops, sorry. Okay, so as we follow this down, right, let's follow this helix, right? 5' to 3'. And this is where this term comes in, anti-parallel. They're parallel with each other, but they run in opposite directions. So the opposite helix goes 5' to 3', but it does so running from bottom to top. Okay, so do you see that? 5' to 3', top to bottom, and the opposing helix is 5' to 3', bottom to top. Okay, and again, the bases stick towards the inside. So here we have a purine, here's a pyrimidine, right? Here's a purine, purine, pyrimidine, okay? And they're stabilized by hydrogen bonding. So remember I said there's no connection. That's true. There's no true bond between the bases, but there is hydrogen bonding, which is a, let's call it a pseudo bond. It's not, they only last for a little while. They're much easier to break than regular bonds. And then here's just another view, another way of looking at it, right? And do you need to memorize this? The amount of space in a turn, etc. No, you do not. Okay. But notice here the hydrogen bonds. Right here, there are 2. Here, there are 3. 3, 2. So 2 hydrogen bonds between thymine and adenine, and 3 hydrogen bonds between guanine and cytosine. So from, because G always pairs up with C. And that's why with Chargaff's rules, you get that. That's how Chargaff's rules came to be. Or why you have equal amounts of C and G and equal amounts of A and T. Then here's just another view of it as well. Okay, complementarity of bases. A forms 2 hydrogen bonds with T, 3 hydrogen bonds between G and C, and that's going to give a consistent diameter. What do we mean here is that so this is a purine, and that's always going to be connected to a pyrimidine. So purine and pyrimidine, because this is a 2-member ring, is always going to be connected to a 1-member ring. 2-member, 1-member. Purine, pyrimidine. And it just so happens that there's 3 hydrogen bonds between these two, and there's 2 between these. The reason for that being is actually just because of the availability of nitrogen and oxygen, because it takes oxygen or nitrogen to make a hydrogen bond, and you have 1, 2, 3 that are exposed here with hydrogens, okay? And so they're available for hydrogen bonding. Whereas over here, or down here, with these, there's only 2 that are available for hydrogen bonding that are exposed. Okay, let's take a look at DNA replication. So after the, quote, discovery of DNA, which was in 1953, if you recall, it was still debated, how does DNA actually replicate itself? Remember, that is a condition that has to be met for the hereditary material. So there are 3 possible ways that DNA could copy itself, and they're called conservative, semi-conservative, and dispersive. So let's take a look at what would happen if any of these were correct. In a conservative model, here is the original double helix. It's represented here as being red. Okay? If, when the DNA copies itself, remember, you have 2 strands, it's double-stranded. If each strand makes a copy of itself, and then the original, quote, parental strand gets back together with the other parent strand, then you would have this. You would have, after the first round, the original parents back together again, and you would have the 2 strands that are the offspring, the daughter strands, what we call them. So the 2 daughter strands get together with each other. Then, after a second round of replication, the original parental strands stay together, and that would be the same for each, right? So you would have 3 blue molecules of DNA and 1 red, representing the original parent. In a semi-conservative model, here's the parental strand, okay? So the original 2 parental strands. So they separate, make a copy, and then they get together and go back into a double helix with the new copy. So after the first round, you have a parent strand with its copy and a parent strand with its daughter strand, or its copy. Now that happens again, okay? So division happens again. What would happen? The original parent strand is with its copy, as happened here up at the top with the other original parent strand, leaving 2 double-stranded DNA molecules that are completely new when compared with the beginning. Okay? That's semi-conservative. And finally, dispersive. What if the 2 parent strands, after making copies, actually did so discontinuously? So what if there was copying and then the strand actually didn't stay together? It joined with the daughter strand. Okay, I know it seems a little strange, but back in the 1950s, this had to be considered as a possibility, right? What if the DNA simply didn't? It dispersed. Okay, so it goes blue, red, blue, red, blue, red, blue, all the way through in each. So you would have this. Now, the parent strand would be dispersed in all of the subsequent daughter strands that are produced, just keeping it, so basically, what that would mean would be that your DNA, you would have some of your original DNA that you had from the time that you were first conceived. When sperm met egg, some of that DNA would still be around. Okay. Well, in 1958, Meselson and Stahl came up with an experiment to figure this out. And again, it involves, as does quite a few experiments, using different isotopes of elements, in this case nitrogen. So DNA has quite a bit of nitrogen in it. And there are actually two isotopes of nitrogen. There's nitrogen 14, which is the more common one, and the one that we find for the most part in the air. So nitrogen makes up most of air. And there is a much, much rarer heavy form, or nitrogen 15. So what they did was they grew E. coli. Remember, E. coli is one of our model organisms. Now we use a model bacterium to do these types of experiments. They grew E. coli in a medium that had nitrogen 15 in it. So when the E. coli grow, and they cultured it, let it grow for many, many generations until all the nitrogen that was in the E. coli was the heavy isotope, nitrogen 14. So then they took these E. coli where all the nitrogen was labeled with the heavy isotope and moved it, switched it to nitrogen 14 medium, or regular nitrogen. So what did that mean? And then they collected samples after every generation to take a look and see, where's the nitrogen 15 going. If they can track the nitrogen 15, they can figure out how the DNA is copying itself. Because the original parent strands would be nitrogen 15, and the new strands would be nitrogen 14. And the conclusion from this experiment was that DNA replication is semi-conservative. And I will show you what it is that they found out. Okay. So first, let's take a look at what's going on here. So here is less than one generation. Okay, less than one generation. They've just put the E. coli into nitrogen 14 medium. Okay, now after one replication, they centrifuge and find out, where is the nitrogen at. If the DNA was conservative, remember here you would have the parent strands, and here you would have the daughter strands. Well, we can rule conservative out right away. Why? Why can we rule that out? The reason we can rule that out is because, well, take a look here. After the first replication, you would have a heavy band of nitrogen 15. So this would be your 15N, and you would have a light band of nitrogen 14. And that is simply not the result. So the conservative model is ruled out right away. Now, what would you expect after 2 generations? If semi-conservative were correct, some of the strands would be half and half, but would include the original parental strands. And 2 would be all new, and that would be your nitrogen 14. So this would be half and half nitrogen 14 and nitrogen 15. So you would have a heavy band and a light band. Now notice right here, this and this, they're the same. So after one replication, you're not able to tell the difference between semiconservative and dispersive because, right, you would have in semiconservative, you would only have two molecules of DNA, and each would have a parent. Whereas in dispersive, it would be all jumbled around. So you would get the same result, and you would have a heavy band. So one generation does not rule out dispersive. It's the second generation that rules out dispersive, because right here in dispersive, you would have you would only have one band, because the original nitrogen 15 would be incorporated all throughout all copies of the DNA. But after two divisions right here, we get two distinct bands. And that is precisely what you would expect from semiconservative replication. And if you go even further with 3, right, you start to see a little bit more separation of the bands, because the original parents are going to start to be outnumbered, right, by new strands. So here was your light, and it would just continue to rise. You would have more and more and more light, where you would only have, it would start to, this band would start to go away after a while, okay? But that is conclusive evidence that DNA replication is semiconservative. So what do we have to have in the replication of DNA? We need to have 3 things, right? Something to copy, okay? So there needs to be a parental DNA molecule. There needs to be something to actually do the copying. And as we've learned, some of the workhorses, the catalysts in our body that do things are enzymes. So it's going to be enzymes that do the copying. And you have to have the building blocks. As we learned, right, with proteins, we have amino acids for our building blocks. For carbohydrates, we have essentially, sugars, right, that are the building blocks. And with DNA, that's going to be nucleotides. Now, there's going to be 3 general steps, 3 general phases of DNA replication. These are going to be initiation, elongation, and termination. And it's just like they sound. So initiation, replication of the DNA starts. So what's going to start it? What's going to be the stimulus that starts the initiation, the replication? Elongation, so new strands of DNA are synthesized. What's going to do that? Remember, enzymes. And finally, termination. Something's going to have to be the signal to end the DNA replication. So if we take a look here, we have, and I'm just going to, okay, here we go. Here's our template strand. And if you recall, right, here's our sugar phosphate backbone. So, right, here we go. Sugar and phosphate, PO4. And that's a repeating scheme going all the way down the strand, and actually 5' to 3', all the way down. Sugar, phosphate, sugar, phosphate. The bases are attached to the number 1 carbon on the sugar, and they stick out like so. Okay, now on the new strand, notice that if you have C, C pairs with G. And remember the 3 hydrogen bonds, H bonds, okay? So C is going to pair with G. So G will have to be synthesized T to A to T, C to G, and on. Now notice here on the template strand, we have A, so something is going to have to bring in this new nucleotide, which notice the nucleotide itself is all 3 things together. It is the base itself, the thymine, we have the sugar, and we have the phosphate, which is going to attach to the OH. Now come over here, and we see that the A is now attached to the T, and the new strand is synthesized going this way. 5' to 3'. That is going to be a repeating, you need to know that. The new DNA is synthesized 5' to 3'. What does that mean? It means that the DNA parent strand, or the template strand, is going to have to be read 3' to 5'. But just remember this, all new DNA is synthesized only in the direction 5' to 3'. And this is getting ahead of us a little bit, but the primary enzyme we're going to look at is called DNA polymerase 3. DNA polymerase 3, which of course would be the 3rd kind of DNA polymerase discovered, because they're labeled in order of their discovery. Okay, so what about this DNA polymerase? What does it do? So the enzyme itself is going to match the DNA bases with the complementary nucleotide and link them together. So it's going to match them up, A with T, and T with A, C with G, and G with C. So all the DNA polymerases, DNA polymerase will see 1, 2, and 3, because we'll be looking at the E. coli model. They're going to have common things to them, okay, to each of them. They add new bases to the 3' end of existing strands. So if you take a look here, right, the new DNA is being synthesized 5' to 3'. This is the template strand being read in this direction, right, with the new bases added on here. So the green is indicating the original parent strand. So the synthesis is 5' to 3', and each of them is going to require this here, this primer of RNA, and that is this red oval, okay? RNA polymerase. Remember, an ASE is an enzyme. So RNA polymerase is an enzyme that synthesizes DNA, okay? So RNA polymerase is going to come in, and that is an RNA primer, okay? And it's going to lay down the initial nucleotides, okay? And then DNA polymerase comes in and finishes it from there, okay? So each of these requires what's called a primer, almost like painting, although nowadays they have paint and primer in one, but it used to be you had to prime before you painted. So thus you get the primer, it gets it ready. Now, when we look at prokaryotic replication, remember I said this, E. coli is a perfect model. And, you know, a bacterium is a good thing to look at because it only has 1 chromosome, and that chromosome is a circle. So the replication is going to begin at one place, which is right here, called the origin, and it's going to proceed in two directions. So the DNA is going to copy in each direction. So now we take a look. Okay, so replication starts, and each of these is called a replisome. What that means is that's just our all of the enzymes that we need are there. We call it the replisome. It's going to require different things, right? A primer, DNA polymerase, and other things that we're going to learn about to get this going. That's just called the replisome. So we're going to have two replisomes, and they're going to proceed all the way around. So notice they're going all the way around, and remember, your new strand is going to be red, correct? So as it goes around, there's a new strand, and an old strand is getting together with a new strand here. And it gets all the way around to the termination point. Now you have the parent strand blue and the daughter strand red. Okay, so it goes around, and you're going to have two. And remember cell division. These are each going to go to the new cells. Continuing with E. coli as our model organism, we now take a look at DNA polymerase. Now, DNA polymerase is the primary enzyme responsible for synthesizing DNA. So it's going to create, the continuous strand of DNA. Now, there are three of them in E. coli, and I'm going to start with number 3, and that is DNA polymerase 3. This is really the primary one, but of course it was discovered third. It's the primary one. It's going to add nucleotides going 5' to 3'. That is synthesizing 5' to 3'. There's also DNA polymerase 1, which is going to act on the lagging strand. We'll see that in a moment, to remove primers and replace them with DNA. And DNA polymerase 2, which is involved in fixing errors in DNA. Now, all of these polymerase are going to have proofreading activity, and that is 3' to 5' proofreading activity. So remember, the DNA that's being copied, the strand that's being copied, is being read 3' to 5'. The new strand is synthesized 5' to 3'. Do not forget that. So DNA polymerase is going to start at a section right here that's called an RNA primer. And we're going to get more into this. You're going to get a better picture in a moment, but think of the two strands being separated. All right, so we have to begin somewhere. So the copying has to begin somewhere. And that's going to begin with an RNA primer that is laid down by an enzyme called DNA primase. And what will happen, notice here's the strand that's being copied, being read 3' to 5'. DNA polymerase will start adding nucleotides as you go along, synthesizing a new strand in the 5' to 3' direction. But of course there's more. Here's 4 other players in the game, and they are all enzymes. Well, except for the SSBs at the end. But they're all proteins. The first one's called DNA primase. I just mentioned this. It's going to synthesize the RNA primers that shows where replication is to begin. Helicase, or DNA helicase, binds to the DNA and it travels 5' to 3', breaking the hydrogen bonds between strands. So what it is responsible for is unwinding the DNA, unwinding the double helix. The third is DNA gyrase. Okay, DNA gyrase, which is, depending on what you're reading, it's a type of what's called a topoisomerase. And this is found in E. coli. Remember, we're looking at the E. coli model for simplicity. So the DNA gyrase is going to relieve stress ahead of the replication fork, or where the DNA is opening up to begin copying. You'll see that in a moment. And finally, single-strand binding proteins, or SSBs, they're going to keep the strands open. They're simply going to be there to keep the strands open so that the base pairs, so that the bases don't want to come back together and hydrogen bond together. And it's probably easier if we just take a look at this. So right here, remember again, we're looking at E. coli. Okay, so remember, E. coli began copying at one place, and we have these two replisomes, and they're continuing, right? They're continuing to copy, this one going in this direction and this replisome going in this direction. Well, what's going to happen as you, almost like an old school phone cord, notice how it's opening up here and it's uncoiling these two strands of DNA here. Well, ahead of that, because of the uncoiling, it's going to cause kinks. It's going to cause the DNA to kink up ahead. So the replisomes, as they go, and oh yeah, that's called supercoiling. So there's your supercoiling. We have an enzyme right here called DNA gyrase, and this is the one that is going to relieve the tension. So DNA gyrase relieves tension. All right, so let's take a look. Here's a good, well, I'm going to take this because I got this from another source. We'll just cross topoisomerase out here. We'll call this gyrase. So here's an enzyme called helicase. The helicase is responsible for unzipping the DNA. Okay, so this here, don't forget this area is called the replication fork, where the DNA is unwound. Now, over on the other end, imagine this end coming around. I'm not drawing a good, there we go. This is going to be unwinding over here, and you're also going to have another replication fork going this way. Because remember, in E. coli, in the circular chromosome, it's going to go both ways. So here we go. The helicase, we'll call this number 1. The helicase is going to unwind the DNA. Number 2, the single-stranded binding proteins are going to keep it stable. Notice they're over here as well. It's going to keep the bases from wanting to come back together and hydrogen bond. Okay. Now we need a place to start. So primase is going to come in and it's going to lay down this RNA primer. So it's made out of RNA, which is single-stranded, but that doesn't matter because we're just dealing with one strand right now. So it's going to lay down a few bases that are complementary to these DNA bases to get the whole thing started. And on we go. And ahead of all this, DNA gyrase is going to be relieving the tension ahead of the replication fork. This is going to lead to a little bit of a problem because remember, we have to synthesize both strands of DNA. And also a good example of why evolution is not perfect. There is a leading strand and a lagging strand. And the idea behind this is that DNA polymerase only adds nucleotides in a 5' to 3' direction. It is not capable of moving in the 3' to 5'. So for the leading strand, which we'll see the leading and lagging strand in a moment, for the leading strand, that's fine. The DNA is going to be synthesized. There's only one primer needed. And as the DNA unzips, it's just going to one primer, and then DNA polymerase 3 will be able to go ahead and synthesize nucleotides all the way down the strand. But on the lagging strand, it's going to have to be synthesized in segments. And these are called Okazaki fragments. Okazaki fragments right here. Okay? So it's going to be synthesized 5' to 3', but in pieces or Okazaki fragments. So these fragments are going to be the RNA primers plus DNA. So maybe better to go ahead and take a look at this. So notice here, on this side we have the lagging strand, and on the bottom half we have the leading strand. So let's start here. This is really one. These are in steps, okay? So step one, the helix has to open up and replication has to start. Okay, let's go forward in time. 2. Here's the leading strand. So here's our RNA primer that gets laid down. Now the DNA can be synthesized going this way, and this can continue all the way down as it goes, okay? Which is what we see here in 3, right? Here is the leading strand. So this is like point in time 2, point 1, 2, 3. Okay, so this is as time goes by. So here's your RNA primer that was laid down right here. And in the leading strand, it's continuous. So the DNA nucleotides are added on one at a time, and it just keeps going. But here, with the lagging strand, notice what happens. It can only synthesize 5' to 3'. So remember, the DNA then is going to be read 3' to 5'. So as the replication fork opens up further, we need an RNA primer laid down, and the synthesis is going to continue this way. Now look through after some time. This was the same primer, first RNA primer. Now the DNA has unzipped even further, but it has to be synthesized in this direction, going up. So a new primer is laid down and the DNA synthesized, okay? The nucleotides are added until it gets to the first RNA primer. So this right here, I'll circle this one. This is what is known as an Okazaki fragment, or discontinuous. So it's going to take many primers along the way for this strand to be synthesized. You'll probably want to come back and take a look at this slide numerous times to get a handle on this. Okay, then finally, there's another enzyme called ligase, or DNA ligase. And this is the enzyme that's going to seal the gap between Okazaki fragments and make them into one long continuous strand. So it comes back and seals the fragments up. Let's see how that works. So here at the bottom is the leading strand. So we're not concerned with that. We know that this one is going to be synthesized all the way. Okay? So this would be, right, reading 3' to 5'. So I should, let me put that in here. This strand down here is 5', and this is 3' here. So this DNA has to be read 3' to 5'. So here is your primase that comes in, and the primase lays down the primer. It's also going to come forward and lay down another primer and another primer. Now here is DNA polymerase 3 that came in. Okay, we don't see it here, but it came in and it synthesized this piece and this piece. And it's then going to come in and synthesize in this open area here, okay, to complete this Okazaki fragment. So there'll be an Okazaki fragment here. Here's one here, and here's another one here. Now, DNA polymerase comes in, okay, and DNA polymerase 1 is going to replace, so DNA polymerase 1 replaces primer. Let me just put RNA primer with DNA. Okay, that's what DNA polymerase 1 is here to do. It is here to replace this primer, so now you're just going to have continuous DNA. But that's not enough. The DNA ligase, so because it still is not connected properly here. So it used to be RNA, now it's DNA, and so you need a link right in here, okay? That's where, and you're going to need a link here and here and here and eventually here, okay? So on either end of these RNA here, on either end of these RNA primers, and that's what DNA ligase is going to do. It's going to connect DNA from DNA polymerase. Oh, shoot. Oh, man. Sorry about that. So the DNA ligase is going to come in. Let's just put this back here. This used to be a primer. So DNA ligase is going to connect DNA from DNA polymerase 1 back together. Okay. So, just in case it wasn't confusing enough, let's take a look and again, just pretend this is E. coli, right? You're just going to have one origin of replication on one end of the circular chromosome. And the forks are going to go in each direction. So this creates a strange situation. So remember, the DNA is being read 3' to 5' and synthesized 5' to 3'. So if it starts here and it's moving along, this one is going to be the leading strand going in this direction. And as the replication fork opens up, we're going to have Okazaki fragments that are built here, 1, 2, 3, until they get to the origin of replication. But once they get there, that turns into the leading strand. So there's each strand can be both a leading strand and a lagging strand from the origin of replication. Okay? So as it moves this way, it's going to be a leading strand, but as it goes this way into this replication fork, it's going to lag, and you're going to have Okazaki fragments. So just know that there's going to be Okazaki fragments, Okazaki frags on each strand. Okay, whoops, I think we were there before. Okay, so let's take a actually, let's come back to this. All right, let's look at step 1, because this has all the players that are in it. So the replication fork is here. Whoops. Okay, so this is the replication fork. So in this case, in this example, we're moving towards the left. Here is the helicase. The helicase is unzipping the DNA as it goes along. Okay? DNA gyrase is relieving the stress over here to keep it from supercoiling and keeping the DNA from getting all kinked up ahead of the helicase. Okay? Here is the leading strand. Here's the leading strand. So the leading strand is going to be read 3' to 5'. Okay, so this is the leading strand right here, and the new synthesized DNA is just moving along, and that's being synthesized by DNA polymerase 3, which is our primary enzyme. Okay, that is our primary enzyme. Note the single-stranded binding proteins down here, okay, which are keeping the bases from interacting with each other, okay? So here we go. The DNA polymerase 2 is just going to march along this strand after it had a primer back here somewhere, right? And it just keeps going. Here is your lagging strand. So it's being read 3' to 5'. So now back of the helicase, the primase lays down a primer right here. Okay? And notice, right, so we got our lagging strand. Here's another RNA primer. Now go back further, and here is DNA polymerase 3. And it is moving in this direction, and it is synthesizing new bases. So it's going to synthesize new bases. It started here, right? You had your RNA primer, and the DNA polymerase 3 is going to march along until it gets to the next primer. So this here is an Okazaki fragment that's being synthesized. Now let's go to the next step. So the loop in the lagging strand allows replication to occur on both. So here we go, right? Here's your DNA polymerase 3. It's marching along. This loop is growing because the DNA is unzipping. And the second Okazaki fragment is nearing completion. So here was the first one. Now here's the second one, and our player, our major enzyme, whoops, DNA polymerase 3. Okay? Now let's look further. When the polymerase 3 on the lagging strand hits the previously synthesized fragment, it releases the beta clamp. I'm not really concerned about that. And the template strand. DNA polymerase 1 okay, so here's your polymerase 3. It's detached. Here is an Okazaki fragment. And then here comes DNA polymerase 1, which is going to replace this yellow primer. Remember, that primer is RNA. It's going to replace the RNA with DNA. And then after a while, it will move over here to do the same on this primer. And then after a while, it's going to come and do that on this primer. Okay, so the clamp loader attaches the beta clamp, transfers this to polymerase 3, creating a new loop in the lagging strand. DNA ligase comes in. So DNA polymerase 1 detaches, and DNA ligase patches it up. Okay? So the ligase is going to patch up that. This only needed to be done one time on the leading strand here. Okay. Now back comes DNA polymerase 3. DNA polymerase 3 is now at work synthesizing the new strand, and it just keeps going like this. It'll go like this, then it'll come back here, and as the DNA unzips, it just keeps going this way. Next, we take a look at eukaryotic replication. Thus far, we looked at our model of E. coli with its DNA polymerases and its associated enzymes that are all associated with replication. But in eukaryotes, definitely more complex, right? Complicated by the larger amount of DNA, multiple chromosomes, right? So we have 46 chromosomes to worry about. And our chromosomes are linear. They're not circular like DNA. So remember, with DNA, you had one origin of replication and one termination site. Great. What do you do if your chromosome is one long piece? Well, thankfully for us, as it says here, the basic enzymology is similar. We're not going to go into the details of all the enzymes that are needed for us, for humans. Our E. coli model will be enough to deal with, really. And if you want to learn more about that, then when and if you take cell biology, I'm sure you'll be glad to learn them all. There are more and different DNA polymerases for eukaryotes, but it is very similar to what happens in E. coli. So again, multiple origins of replication are going to be needed because there is so much more DNA to be copied than there is with bacteria. They say bacteria, 80 to 90% of the DNA in a bacterial chromosome actually codes, are actually genes. So there's only 10 to 20% DNA that does not code for anything. Not so with eukaryotes. There's much more what we might call junk DNA that doesn't code for anything. Okay, so we're not quite sure exactly why there's multiple origins of replication, except for speed, but where exactly they happen, not quite sure that we know that. There are more factors to assemble helicase and primase complexes, then load up the polymerase with that sliding clamp unit that you saw. So there's just more involved in eukaryotic replication. So let's take a look back here at our E. coli. So we had one origin of replication, right? The DNA unwinds. We get this replication bubble and two replication forks, one on each end. So here we go. Here's the forks, and synthesis continues in both directions. And here's a kind of a blow up looking at the bubble itself and how synthesis, the copying, is going to happen in two directions, right? So here was the origin of replication, and it goes and it just keeps going in both directions until it gets all the way around to the termination site, at which point you have two copies of the circular chromosome. Now, that was our E. coli model. In eukaryotes, we have linear DNA. So we have one long continuous strand. So what's going to happen is you're going to have different origins of replication all the way along the chromosome, which are indicated with the blue here. So the DNA unwinds, and it begins at multiple origins of replication. So if you can imagine this one right here, continuing in this direction, it's going to keep going. It's going to have a leading strand and a lagging strand. And it'll keep going, but this one's going to be going in the same direction until they meet and the copying is done. And that happens along the entire length of the chromosome. So now you have, right, remember semiconservative replication. Here is a parent strand with its newly created strand rebound together, and we can see the kinetochore proteins at the centromere of the chromosome that are, associated with the centromere region, and we have a nice replicated chromosome. So, I'm not going to go over this step by step in too much detail, but it's here in words for review, right? DNA unwinds. The DNA helicase opens up the DNA and continues in two directions. The SSBs, the single strand binding proteins, coat the DNA, keeps the bases from coming back together. DNA gyrase works ahead of the fork to keep supercoiling from happening and all the kinks in the DNA. The DNA primase lays down an RNA primer. DNA polymerase 3 adds nucleotides in the 3' OH region. Okay, so adds nucleotides to the 3' OH end of the primer. And then elongation of both the lagging and the leading strand continues. The leading strand continuous, the lagging strand made of Okazaki fragments that are done discontinuously. The RNA primers are then removed and they're replaced with DNA by DNA polymerase 1. And then finally, the DNA ligase comes in to seal that gap with a phosphodiester bond. And it helps with those, if you remember back a ways in this lecture. Will help in forming those phosphodiester bonds in the sugar phosphate backbone. Another problem with eukaryotes is telomeres. Telomeres are essentially the ends of the chromosome. And telomeres, as it says, protect the ends of the chromosome from nucleases, which are enzymes that can degrade DNA in the nucleus, and to maintain the integrity. Now, this is, again, the ends of the chromosomes, which they're going to have a special problem. And that is that on the lagging strand with the Okazaki fragments, the primer cannot be replaced at the very end, just on the lagging strand. So the chromosomes eventually become shorter and shorter. So, as you might imagine, you cannot have genes down there. So there are really no genes down at the very ends of the chromosomes, because they're going to become, and they would become, shorter and shorter and shorter with every division. That's a problem. It's a big problem. So here we go. Let's take a look. Here's the leading strand. Like it says, no problem, right? So here it is. It's being read 3' to 5', and your new DNA is being laid down all the way to the end. The lagging strand is going to have a problem, okay? And as you see here, let's look at the, here's your leading strand, here's your lagging strand. And the problem is right here with this primer, which when we blow that up, that's this area here. it can't be replaced. It cannot be replaced. That's a big problem. So one would hope that this DNA does not code for anything. And luckily it does not. Okay, so primer is removed. So what happens? It cuts off the whole thing. Okay, so the template has now been shortened. If this just kept going on in a rapidly dividing cell like skin cells, you would, over the years, run into a lot of problems with your chromosomes getting shorter. So luckily the telomeres have these short repeat sequences of DNA that are actually synthesized by an enzyme called telomerase. And what this does is it makes the telomere section of the lagging strand using an RNA template, okay? Not the DNA itself. Again, the leading strand can be replicated all the way to the end by DNA polymerase 3. Not so with the lagging strand. And also, great example of how evolution is not perfect in any way. This seems very, very inconvenient, and it seems like a evolutionary answer for it. Telomerase. To have an enzyme to take care of this and just essentially to insert DNA that does not code for anything, so that when it gets cut off, you're not losing genes. Okay? There is a relationship between the activity of the telomerase and senescence, or old age. So as we age, telomerase activity seems to go down, and thus the length of our telomeres go down, and they become more brittle, literally. So with older age, our telomeres begin to shorten. In youth, it's not a problem, and this is an active area of investigation. Why is it that telomerase activity goes down? Because this is it is a good solution to the problem, having this enzyme, but why it decreases with age, we don't really have an answer for that. We do not. But let's see how it works. So to synthesize something with telomerase, so here we have the strand, and we have these 3 nucleotides in our removed primer. Okay, so this is going to get added in. This whole big oval is telomerase, and part of the telomerase is RNA. So there is an RNA template that has these strange repeating sequences. In this case, we can see AA, CCCC, AAC. And it's going to catalyze the synthesis of complementary DNA nucleotides that are going to come in. And this is going to keep the telomere, keep the telomere extended in this way. And this enzyme moves along, and it just keeps putting in these repeats, so that when we get the Okazaki fragments in the lagging strand, we're just cutting off junk. Okay, and it gets resynthesized. And again, the activity of this enzyme goes down as we age, and literally our chromosomes start to shorten. And it's hypothesized that this is one reason for old age, for the effects of old age, and eventually death. Okay, what about repairing DNA? Because this has to be something I haven't mentioned. This has to be a very, very, as they say, high fidelity activity. You cannot afford to have your DNA copied improperly in regions that are coding for something, also known as genes. Cannot really afford to have errors. And there are errors. There's billions of base pairs and bases, billions of nucleotides in our genome. Errors do happen. So we hope that they're caught. The first line of defense is the DNA polymerases. So they have proofreading ability. They can go back. So remember the DNA polymerase 1. There was the one that wasn't mentioned, DNA polymerase 2, which was a proofreading polymerase that can go back and actually make repairs. But we found, scientists have found even more to highlight how important it is that your DNA is copied correctly when cells divide. So to repair DNA, it's going to fall into two categories, specific and non-specific repair. So I'm going to have one example for specific repair. Targets a single kind of lesion in DNA, a lesion being an error, essentially, and repairs only that damage. So we'll look at one type coming up. And also non-specific, a single mechanism that repairs multiple kinds of lesions in DNA. One of those would be DNA polymerase 2. But first let's look at specific. This is a specific mechanism called photorepair that is done for damage by UV light. We are susceptible to damage from ultraviolet radiation. And UV light, ultraviolet light, can cause what are called thymine dimers. Okay, thymine dimers. And this is between adjacent thymines, T's, in the genetic code. So where you have two T's together, there can be a problem between them in that they're covalently linked together. Remember that the bases are not linked together. They should be hanging free off of the phosphate sugar backbone. Sometimes between thymines, UV light can cause them to be covalently linked. That's a problem. In comes an enzyme called photolyase, which it's a good name, photo having to do with light, lyase. So this actually absorbs light in the visible range, and it can use the energy from light to break the dimer between the two thymines. Let's take a look. So here's DNA with adjacent thymines, right? So here's your strand, your parent strand had two A's, and in the new copy, that's gonna, the complementary base is thymine. So UV light comes in, and it can cause this covalent bond to occur between the thymines. Now they cannot hydrogen bond with the adenines that are straight across from them. That's a problem. So photolyase comes in, and it actually does cause this little kink in the DNA because they're no longer free on the backbone. So this enzyme comes in, and it can actually break that covalent bond and allow the hydrogen bonding to occur between adenine and thymine. So that is an example of a specific repair that can happen with DNA. So there's also excision repair. So this is where you have a region that's damaged, okay, and it's literally removed, taken out, and replaced by new DNA synthesis. So perhaps it simply is the wrong base that was put on. So this is going to involve 3 steps. First, there has to be recognition that there is damage. There has to be removal of that damaged region, and then it has to be resynthesized using the information on the undamaged strand as a template. So let's take a look. Now in this case, let's just assume that the base right here is C. And so C equals cytosine. And let's say that the incorrect base that was put in was C, or another cytosine. They do not interact. Okay? Remember, cytosine is a pyrimidine. Okay? We need to have, so need G to match up with C. So we have an enzyme that comes in, not really concerned that it's called the UVR-ABC complex, but it comes in, and this enzyme will actually cut out 3 base pairs and take them away. What does that allow? That allows DNA polymerase, in this case let's just call it DNA polymerase 2, to come in and actually lay down the correct bases. Don't know what this one is. We'll just put asterisk, and it's going to put in a G, and it's going to put in the correct base here to go along with the original parent strand. Okay, so now we have C and G, and they're able to bond together, hydrogen bonding. Okay, that is it for this DNA presentation. If you have any questions on it, you can ask me, or go back and replay. I know this is a very confusing topic with a lot of enzymes, so you might need to go over this a few times. Okay, thank you very much for watching.